This slide explores the expression of Prame, EZH2, and rare isoforms in both patient samples and cell lines, aiming to uncover their potential role in the biology of soft tissue sarcomas and their subtypes. Panel A shows in silico mRNA expression in soft tissue sarcoma samples. Prame and EZH2 mRNA levels are significantly higher in synovial sarcoma compared to other soft tissue sarcoma subtypes like leiomyosarcoma and rhabdomyosarcoma. Among the rare isoforms, rare A and rare B show significant differences across subtypes. Rare gamma expression does not significantly differ. Panel B mentions that Prame expression is positively correlated with disease progression upper panel and negatively with survival outcomes lower panel. Patients with amplifications in Prame, red dots, tend to show worse clinical outcomes compared to those with diploid or shallow deletions. This highlights Prame's potential as a biomarker for disease progression and prognosis in synovial sarcoma. In Panel C, synovial sarcoma clusters are divided into three biological subgroups. Firstly, synovial sarcoma type 1, poorly differentiated, which tend to have variable Prame expression. Secondly, synovial sarcoma type 2, monophasic, and thirdly, synovial sarcoma type 3, biphasic, where Prame expression shows observable trends but is not significantly different across clusters. Prame's expression does not strongly differentiate among synovial sarcoma cluster subgroups, but it remains relevant as a biomarker. Panel D, where we see mRNA expression in soft tissue sarcoma cell lines, shows that synovial sarcoma cell lines, example given CO1, have elevated Prame and EZH2 mRNA levels. Rare isoforms, particularly rare B, show significant differences between soft tissue sarcoma cell lines. This analysis demonstrates that Prame and EZH2 remain consistently expressed across both patient samples and cell lines. In Panel E, we see protein expression analysis. The key observation, as Western blot shows, are that Prame and EZH2 proteins are expressed in translocation-positive synovial sarcoma cell lines. The fusion protein SS18SSX is exclusive to translocation-positive synovial sarcoma cell lines, example given CO1 and HSSY type 2. GAPDH and B-actin validate equal protein loading. This confirms that Prame and EZH2 are not only transcriptionally active, but also translated into functional proteins in translocation-positive synovial sarcoma. In summary, Prame and EZH2 are consistently elevated in synovial sarcoma and correlate with worse clinical outcomes. Protein validation highlights the translational relevance of these genes. Rare isoforms might provide additional insight into soft tissue sarcoma subtypes. Targeting Prame and EZH2 offers a potential therapeutic avenue for synovial sarcoma treatment. This slide explores how Prame expression correlates with gene expression, chromatin remodeling, and cell cycle regulation in synovial sarcoma specimens. Prame is a known cancer testis antigen with critical roles in oncogenesis and immune responses. Researchers aim to understand the molecular pathways and gene expression profiles associated with varying levels of Prame expression to identify potential therapeutic targets and biological insights. Panel A shows a volcano plot illustrating the correlation of gene expression with Prame levels in synovial sarcoma specimens. The x-axis represents the log-fold change of gene expression normalized to Prame, while the y-axis shows the adjusted p-values. Red dots highlight gene positively correlated with Prame expression, while green dots represent negatively correlated genes. This analysis reveals distinct groups of genes that are differentially regulated depending on Prame expression levels, suggesting their potential involvement in Prame-mediated pathways. Panel B represents a heat map of the top 20 differentially expressed genes between Prame high and Prame moderate samples. The median expression score was used to separate the groups. Prame high expression samples are labeled in dark green, while Prame moderate samples are in light blue. The heat map showcases how gene expression profiles differ significantly with varying Prame expression levels, underscoring the relevance of these genes in Prame-related pathways. Panel C shows GSEA results, identifying pathways enriching Prame high versus Prame moderate expression. In the first plot, the rare bound gene set has a normalized enrichment score of -146, indicating negative correlation with Prame expression. In the second plot, the chromatin remodeling gene set is positively correlated with a normalized enrichment score of 1.699, showing that Prime high samples are enriched for genes involved in chromatin structure changes. In the third plot, the cell cycle checkpoint gene set also shows a positive correlation with a normalized enrichment score of 1.772, suggesting Prame's involvement in cell cycle regulation. Each green curve represents the enrichment score, calculated as the running sum of genes rankings from Prame high to Prame moderate. Adjusted p-values confirm the statistical significance of these pathways. Panel D summarizes the gene ontology pathway analysis, with enriched pathways representing along the z-axis and their corresponding gene ratios on the x-axis. The size of the dots reflects the number of genes enriched in each gene ontology term, while the color gradient indicates significance, adjusted p-values. Key enriched pathways include leukocyte-mediated immunity, extracellular matrix organization, and chromatin remodeling, suggesting diverse biological processes influenced by Prame expression. The key takeaways are that Prame expression is associated with significant changes in gene expression, including those involved in chromatin remodeling and cell cycle regulation. Pathways like RAR signaling, immune regulation, and chromatin remodeling could serve as potential therapeutic targets in Prame-high cancers. This multi-phased approach combining GSEA, gene ontology analysis, and differential gene expression reveals a comprehensive picture of Prame's role in synovial sarcoma specimens. This slide investigates the role of the SS18-SSX fusion protein in regulated Prame expression in synovial sarcoma cells. The analysis integrates chromatin immunoprecipitation, RNA, and protein studies to highlight the molecular mechanisms. Researchers aim to demonstrate how SS18-SSX interacts with the Prame promoter and influences its expression, highlighting its role in the oncogenic process of synovial sarcoma. Panel A demonstrates chromatin immunoprecipitation analysis of SS18-SSX binding to the Prame promoter. In CO1 and Mojo cells, SS18-SSX binding to the Prame -66 to +35 promoter region, represented in red, is significantly enriched compared to controls, isotype and irrelevant regions. Positive control, the blue, validates the assay, while the negative control, green, confirms specificity. This suggests a direct interaction of SS18-SSX with the Prame promoter, driving its transcriptional activation. Panel B examines the effect of SS18-SSX knockdown on Prame expression. Cells treated with cRNAs targeting SS18-SSX1 or SS18-SSX2 show a reduction in Prame protein levels, as visualized via Western blot. GAPD8 serves as the loading control, ensuring consistent protein quantification. Panel C quantifies the knockdown experiments in Panel B. Both CO1 and Mojo cells show significant reduction in Prame expression following SS18-SSX knockdown. This quantification confirms the regulatory role of SS18-SSX on Prame expression. Panel D explores the effect of ectopic expression of SS18-SSX in translocation-negative SW982 cells. Overexpression of SS18-SSX leads to a marked increase in Prame mRNA levels, indicating that SS18-SSX is sufficient to drive Prame transcription in non-translocated cells. These results are consistent with the hypothesis that SS18-SSX transcriptionally regulates Prame. Panel E confirms that ectopic expression of SS18-SSX also increases Prame protein levels in SW982 cells. GAPD8 serves as a loading control, and molecular weights are indicated. This provides further evidence of a direct regulatory link between SS18-SSX and Prame expression. The key takeaways from this slide are that SS18-SSX binds directly to the Prame promoter, activating its transcription in synovial sarcoma cells. Reduction of SS18-SSX leads to a significant decrease in Prame expression, confirming its regulatory role. Expression of SS18-SSX in non-translocated cells is sufficient to upregulate Prame, further supporting the mechanistic link. In conclusion, these findings establish SS18-SSX as a transcriptional regulator of Prame in synovial sarcoma, revealing potential therapeutic targets for disrupting this oncogenic axis. This slide explores the interaction between Prame, EZH2, and RARA in CO1 synovial sarcoma cells under different treatments, including GSK343, ATRA, and their combination. The aim is to analyze how these treatments affect protein-protein interactions, histone modification, trimethylation of H3K27, and expression levels of K players in oncogenic signaling. Panel A uses a proximity ligation assay (PLA) to visualize and quantify the colocalization of Prame and EZH2 upper panel and Prame with RAR-A lower panel. Green fluorescents represent PLA signals indicating protein-protein interactions, while blue (JAPI) marks nuclei. Treatments include DMSO control, GSK343, SH2 inhibitor, ATRA, retinoic axing, and their combination. In the Prame-SH2 interaction, the combination treatment significantly reduces PLA signals compared to controls, suggesting disrupted interaction. For Prame-RAR-A, no significant differences are observed, indicating this interaction is less treatment sensitive. Panel B quantifies the PLA signals per cell for Prame-SH2 top and Prame-RAR-A bottom. The violin plots reveal a significant decrease in Prame-SH2 interaction signals with GSK343 and the combination treatment compared to DMSO. Prame-RAR-A signals remain largely unchanged, consistent with the imaging results in Panel A. One-way ANOVA confirms statistical significance with p-values marked as p lower than 5%. Panel C shows Western blot analysis of key proteins, SH2, Prame, and RAR-A, and trimethylation of H3K27 after 72 hours of treatment. GAPDH serves as a loading control for SH2, Prame, and RAR-A, while total H3 normalizes trimethylation of H3K27. The combination treatment markedly reduces H3K27 trimethylation levels, indicating effective inhibition of SH2's methyltransferase activity. Levels of SH2, Prame, and RAR-A remain consistent across treatments, suggesting that the effects are more functional than related to protein abundance. Panel D quantifies the protein expression data from Panel C. The left three graphs confirm no significant changes in SH2, Prame, or RAR-A protein levels across treatments. The rightmost graph highlights a significant reduction in H3K27 trimethylation levels with GSK343 and the combination treatment, confirming SH2 inhibition. These results reinforce the idea that GSK340 and ATRA synergize to disrupt SH2 function, rather than altering protein levels directly. In conclusion, GSK343 and combination treatments reduce the colocalization of Prame and SH2, potentially altering oncogenic pathways. The combination treatment effectively reduces H3K27 trimethylation levels, highlighting the functional impact of SH2 inhibition. Prame, SH2, and RAR-A protein levels remain unaffected, emphasizing the mechanistic focus on functional, not abundance-related changes. These findings suggest that targeting SH2 with GSK340, especially in combination with ATRA, disrupts Prame-SH2 interactions and reduces oncogenic histone modification, offering insights into potential therapeutic strategies. This slide investigates the effects of GSK343, ATRA, and their combination on cell viability, colony formation, and Prame expression in synovial sarcoma cells, including Prame knockdown experiments to assess the role of Prame in treatment response. Researchers aim to understand how Prame impacts the efficacy of GSK343 and ATRA treatments in synovial sarcoma, focusing on cell proliferation, colony formation, and protein expression. Panel A shows the relative cell number after 7 days of treatment with DMSO control, GSK343, ATRA, or the combination combo. In CO1 and Mojo cells, the combination treatment significantly reduces cell viability compared to single treatments, with the pre-GSK343 plus combo approach showing additional effects. For HSSY type 2 cells, only modest effects are observed, while SW982 cells are resistant to treatment. Statistical analysis confirms significant reductions. Panel B examines the long-term impact of treatments on the colony-forming ability of CO1 and Mojo cells. GSK343 and ATRA alone reduce colony formation to varying degrees, but the combination treatment completely abolishes colonies in both cell lines. These results highlight the synergistic effect of GSK343 and ATRA in suppressing cell proliferation over time. Both Panel C quantifies the colony formation data from Panel B. In both CO1 and Mojo cells, the combination treatment significantly reduces colony numbers compared to single treatments, with results consistent across five independent experiments. Statistical analysis confirms the robustness of these findings. Panel D shows Prame protein levels in CO1 cells following knockdown with two different shRNA constructs, SH-Prame1 and SH-Prame2, and treatment with ATRA or DMSO. Knockdown of Prame reduces its protein levels regardless of ATRA treatment, confirming the efficacy of the shRNA constructs. This establishes the experimental framework for subsequent functional analysis of Prame knockdown. Panel D tracks the proliferation of CO1 cells with or without Prame knockdown, SH-Prame1, SH-Prame2, under DMSO or ATRA treatment. Control cells show significant growth inhibition with ATRA, whereas Prame knockdown cells exhibit resistance to ATRA. These results highlight the critical role of Prame in mediating the response to ATRA treatment, suggesting its potential as a therapeutic target. In conclusion, the combination of GSK3-4P and ATRA significantly reduces cell viability and colony formation in synovial sarcoma cells, particularly CO1 and MOJO. Prame knockdown diminishes the efficacy of ATRA, confirming its role as a mediator of ATRA sensitivity in synovial sarcoma. Targeted Prame or combining ATRA with EZH2 inhibitors like GSK3-4P could be promising strategies for synovial sarcoma treatment. These findings highlight the importance of Prame in synovial sarcoma treatment and suggest that disrupting the Prame-mediated pathway could enhance therapeutic efficacy. This slide investigates the impact of ATRA, GSK3-4-3, and their combination on retinoic acid-regulated target proteins and RAR-A expression in synovial sarcoma cell lines CO1 and MOJO. The focus is on evaluating protein expression changes and visualizing RAR-A localization to understand how these treatments modulate retinoic acid signaling. Panel A shows the expression of retinoic acid-regulated target proteins in CO1 cells treated with DMSO, ATRA, GSK3-4-3, and their combination. Proteins like P21, tubulin B type 3, and SOX9 show increased expression after treatment, particularly with ATRA or the combination. This upregulation highlights the activation of differentiation-related pathways. Conversely, oncogenic proteins like SEMIC and PPAR-gamma are significantly downregulated, especially after 7 days of combination treatment. These changes reflect a therapeutic shift from proliferation to differentiation. B-actin serves as the loading control to ensure equal protein loading across samples. Panel B visualizes RAR-A expression and localization in CO1 and MOJO cells under different treatment conditions. RAR-A is marked in green, actin filaments, phalloidin in red, and nuclei DAPI in blue. The combination of ATRA and GSK3-4-3 enhances RAR-A nuclear localization as seen in the CO1 cells. This suggests that ATRA drives RAR-A activation, while GSK3-4-3 complements by modulating chromatin accessibility. In MOJO cells, the combination also enhances nuclear RAR-A localization, but to a lesser extent, indicating possible cell line-specific responses. In summary, combination treatment promotes differentiation-related protein expression, example given P21, tubulin B type 3, while suppressing oncogenic markers, example given CMYC, PPAR-gamma. RAR-A nuclear localization, ATRA and GSK work synergistically to enhance nuclear localization of RAR-A, amplifying retinoic acid signaling in synovial sarcoma cells. Targeting retinoic acid pathways with epigenetic modulation, GSK3-4-3, and differentiation therapy, ATRA, could represent a promising approach for synovial sarcoma treatment. In conclusion, this study demonstrates that combination therapy with ATRA and GSK3-4-3 effectively shifts synovial sarcoma cells toward differentiation and downregulates oncogenic pathways, providing mechanistic insights into potential therapeutic strategies. This slide explores the effects of GSK3-4-3, ATRA, and their combination on synovial sarcoma cells, CO1 and MOJO, focusing on differentiation, senescence, and the induction of senescence markers. The goal is to assess how these treatments disrupt the RAR-A Prame EZH2 complex, driving differentiation and senescence. Panel A examines the expression of tubulin B type 3, a marker of differentiation in CO1 and MOJO cells after 3 weeks of treatment. In DMSO-treated cells, tubulin B type 3 expression is minimal. Treatment with GSK3-4-3 or ATRA alone increases its expression modestly. The combination treatment combo shows the highest levels of tubulin B type 3 with well-structured filaments, indicating enhanced differentiation. These results highlight the synergistic effect of GSK3-4-3 and ATRA in driving differentiation. Panel B assesses senescence in CO1 and MOJO cells via B-galactosidase staining after 10 days of treatment. DMSO-treated cells show minimal B-gal staining. GSK3-4-3 and ATRA individually increase staining, indicating some induction of senescence. The combination treatment results in the most pronounced B-gal staining, as seen in enlarged and flattened cells, characteristic of senescence. Insets show enlarged B-gal positive cells in combo-treated samples. Panel C quantifies the percentage of B-gal positive cells from Panel B. In CO1 and MOJO cells, the combination treatment significantly increases the proportion of senescent cells compared to single treatments. Statistical analysis confirms the robustness of these findings, with significance levels marked. Panel D examines the expression of P16, a key marker of cellular senescence using Western blot analysis. In both CO1 and moisture cells, GSK343 and NATRA individually induce P16 expression, but the combination treatment results in the highest P16 levels. B18 serves as the loading control, ensuring consistent protein loading across samples. In summary, the combination of GSK343 and NATRA significantly enhances tubule B type 3 expression, promoting differentiation in synovial sarcoma cells. Combination treatment drives cellular senescence, as evidenced by increased Bigel staining and P16 expression. GSK343 and NATRA work synergistically to disrupt RAR-A Prame EZH2 complex, promoting both differentiation and senescence. These findings underscore the potential of combining GSK343 and NATRA to target synovial sarcoma by inducing differentiation and senescence, disrupting key oncogenic pathways. What are the implications of inducing senescence as a therapeutic strategy for synovial sarcoma? Could this be extended to other cancers? Share your thoughts in the comments and subscribe for more insights into cancer research and novel therapeutic approaches.